So before proceeding, let's discuss some core feature of Oracle. So in Oracle, we provide live classes, recorded classes, notes, and MCQ. Live classes is for to create the learning of the students, where knowledge meets interaction. Then recorded session are also provided for all the first year student, in which you will have the access of all the subjects through recorded session anytime, anywhere. Then also we provide notes with ultimate comprehensive books with CBME based question answer MCQs and full zone for self-assessment. Then also MCQs are there from beginner level to expert level, which contain standard multiple choice questions. Then student can also book a personalized counseling session in which you will get a direct interaction with the counselor. Then we have some subscription plans starting from basic to premium. So basic plan is of 3 months, standard is of 6, and premium is of 12. The premium plan, we include all the services that is provided by Oracle, like pre-recorded classes, live class, live dot clarification, one-to-one personalized guidance, notes, and quick revision examination. And also for instant 20% discount, you can use my code, that is Anchal20. So as we have started the internal marathon series, so these are the core feature of this series to provide the rapid revision of all the important questions, also volatile fact, quick revision. Then also we provide multiple questions that will be covered, and we assure top notch grades in the examination. Now, as we have started the marathon series over the muscle physiology, so we're going to discuss the main and important topic of the muscle physiology. Like we're going to cover the difference between all the muscles, then also sarcomere, sarcotubular system we're going to cover, then role of calcium, and along with that, we're going to cover very important two diseases, that is myasthenia gravis and TED. So first, we're going to start with the classification of muscles, right? So as we know, the human body has more than 600 muscles, right? And muscles are used to perform many useful functions, like they help in doing everything day-to-day activity, right? So muscles are classified as skeletal, cardiac, and smooth muscles. Okay. So depending upon their situation, they are classified like this. And one by one, we're going to know and we're going to understand the functional and anatomical properties of all these muscles, right? So first we have skeletal muscles. So skeletal muscles is situated in association with bone, right? So they are bone forming the skeletal system. And the skeletal muscles constitute 40 to 50% of body mass and they are always voluntary, and they have striated muscles. Okay. Now the fibers of the skeletal muscles are arranged in parallel, and in most of the skeletal muscles, the muscle fibers are attached to tendon or their end. Okay. Now the skeletal muscles are anchored to the bones by the tendons. Now next we have the cardiac muscles. So as the name suggests, cardiac muscles. Cardiac muscles, they are found in heart, right? Now, they form the musculature of heart. Fine. Then next we have the smooth muscles. Now, smooth muscles are always situated in association with the viscera. Viscera means they are situated in association with organs. Right? Now, it is known as visceral muscles because it is different from skeletal and cardiac muscles because of the absence of cross striation. So as we know, both the skeletal muscles and the cardiac muscles are striated, whereas the smooth muscles are non-striated, right? Now the smooth muscles are also supplied by autonomic nervous system, and these smooth muscles form the main contractile unit of the wall of the various visceral organs. Now we're going to discuss the main and important difference between the skeletal, cardiac, and smooth muscles. So as we know, the type of cells found in skeletal muscles are long tube multinucleated cells aligned with near B cells. Then in cardiac muscles, these cells are long branch with single nuclei aligned with near-B cells, and in smooth muscle they are spindle shaped with single nuclei and packed. And already we have discussed that in skeletal and cardiac muscle the muscles are striated, striations are present, whereas in the smooth muscles they are absent. Okay. Now talking about the location of the nuclei, so in both skeleton and the cardiac muscles the location of the nuclei will be central, right? whereas in the skeletal muscles, it will be different. It will be located at the periphery. Now, talking about the T tubules, so the part of the triads at AI junction. This is present, and in the cardiac muscle, it is the part of dyads at ZX, and in smooth muscles, no T tubules are present. Now, talking about the sarcoplasmic reticulum, so as all the cells have endoplasmic reticulum, the muscles have sarcoplasmic reticulum, right? Now, in skeletal muscle, they are abundant with two terminal cisterns in the triads, whereas in cardiac muscles, it is less abundant with one terminal per sarcomere in triads, and there are no distinctive organization is present like sarcoplasmic reticulum. Okay. Now talking about the distinctive structural features, so the skeletal muscles are highly organized sarcomere and also triads are present, whereas in cardiac muscle, the very important feature that intercalated discs are present with adhesion and gap junction, whereas in smooth muscles, gap junction, calculi and dense bodies are present. Okay, now talking about the contraction mechanism, so it has the calcium binding to troponin, and C exposes myosin binding site on actin. That phenomena also we're going to discuss in today's class, that how it occurs, and it is just similar like skeletal muscle that occurs in cardiac muscles, and here in smooth muscle the calcium binds with camudulin, triggers MLK-CK mediated phosphorylation of myosin and actin binding. Now talking about the covering. Okay, so connective tissue, it has endomyosin, perimyosin and epimyosin. And in cardiac muscles, it has endomyosium, subendocardial and supracardial, whereas in smooth muscle, endomyosium and less organized CT sheets are present. So now the next we have the location. So in skeletal muscles, the location are tongue, upper esophagus, eyes, over bones. All these are the location of the skeletal muscles, whereas the cardiac muscles are only present inside the heart, right? Now the smooth muscles, they are present, as we talk about that they are always associated with viscera. So they are present, mostly associated with blood vessels and the walls of some organs, right? Now, talking about the very important thing about the innervation. So the motor, they are skeletal muscles are always voluntary in action, whereas the cardiac muscles are autonomic. That's why they are involuntary, and also the smooth muscles are also autonomic and involuntary, right? Now, talking about the growth and renewal, so it has very limited renewal property, whereas the cardiac muscles has little, and the smooth muscles are also no as such properties present, right? So this is all about the difference between smooth muscles, skeletal muscles, and cardiac muscles. Okay, next we're going to talk about the very important structure, that is sarcomere. Okay, now first of all, what is sarcomere? Sarcomere is defined as the structural and functional unit of a skeletal muscle. Right? Now, it is also called the basic contractile unit of the muscle. So, sarcomere is also known as the basic contractile unit. Okay, so one by one, we're going to talk about all the components of the sarcomere. So, as we can see, here we have the Z line, here and here, right? This is representing the Z line here. Okay, so each sarcomere is extended between two Z line, right, of myofibril. Okay, now each myofibril contain many sarcomere arranged in a series, right? So they are arranged in series. Okay, now when the muscle are relaxed, that time the average length of the each sarcomere at the time of relaxation, average length is 2 to 3 mu. Okay, now we're going to talk about the various components. So as we can see, in sarcomere, we have 1/2 I band, then we have A band, one dark A band we have, then another we have one half I band, right? And in the middle of the A band, we have a light area that represent the H zone, okay? And H also indicate the hell in which means it indicating the Henson, the name of the scientist from which the H zone is given. Okay. Now, in middle of the H zone, we have the another part that is myosin. So now we're going to draw the whole. If this is the actin filament, here we have actin, 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 right? So actin are always the thin filament, whereas the myosin are the thick filament which is present in middle. Okay. Then we have the Z line. Okay, so this is actin. This is myosin filament. Then here we have Z line. And in the middle of the myosin, we have another line that is known as M line. Okay. And here, starting from this to this whole zone is known as A band. And only this particular area is known as H zone. Okay, so basically this M line between it indicating the mitral, okay, means middle, okay, and M line is formed by the myosin binding protein. So this is all about the one sarcomere that we have drawn. Okay, now next we have the muscle protein. Next we're going to talk about the muscle protein. So as we know, the protein basically are the contractile element, right? So myosin filaments are formed by myosin molecules, and the actin filament are formed by the three types of protein, and those proteins are known as actin, tropomyosin, and troponin. Okay, so myosin filament are formed by myosin molecules, and the actin filament are formed by these three proteins. Okay, and all the four protein, actin, tropomyosin, myosin, and troponin together constitute the contractile protein, or they are the contractile element of the muscle. So one we're going to discuss. So first we're going to talk about the myosin. So as we can see here, the different protein name are also. So in the end, we're also going to discuss about all these. So first we have myosin molecule. So what is myosin molecule? This molecule contain about 200, each myosin molecule contain about 200 molecules of myosin, each filament. Okay. Now, if we talk about the parts, so as already we know that the myosin molecule look like this. It has the head, then it has the shorter arm, then it has the tail. So here we have the head, which has the binding side, then it has the short arm, and also it has tail. So this is all about the structure of the myosin. And also each myosin head has two attachment side. One side is for the active filament and the another side is for the ATP molecule. Now, what is the role of ATP molecule? That also we're going to discuss. So please remember, each myosin has head has two attachment side, one for actin and the another for ATP. Okay. Now we're going to talk about the actin molecule. Now, actin molecule are majorly constitute of the thin actin filaments, right? And each actin filament is known as F-actin, and its polymer of a small proteins are known as G-actin. So if we talk about the G-actin, so they are like small protein molecules, okay? And when we talk about the F-actin, so they are like, if they are together going to bind with each other, so they will be forming like this, okay? So these are known as globular actin, right? And F-actin is the combination of all the globular molecules. Then what is actin molecule is 2F-actin is going to bind like this. So they will be forming actin molecule, okay? Okay, so basically the actin molecule is the actin filament, and also they are forming the double helix structure. Okay, so this is forming the double helix structure, right? So next we have tropomyosin. So about 40 to 60 tropomyosin are situated along the double helix. So first of all, we should know that what is the location of tropomyosin. So tropomyosins are very small proteins that are located along over the actin molecule. Okay. Now, about 40 to 60 tropomyosin molecules are situated along the double helix strand of the actin filament. So if I draw the one strand of the actin filament, okay, this is the one strand and this is the second one. Okay, so the actin filament or tropomyosin will be like this: 1, 2, 3, 4, 5, 6, 7, 8. Okay, now what is the function? The function of the tropomyosin to cover the site of binding. Okay, so tropomyosin are always present on the site at which the actin is going to bind with myosin. Okay, now next we have troponin. This troponin is present in 3 subunits. First, we have troponin 1. Then we have uh, troponin T. Then troponin C. So this troponin first is troponin I, then second is troponin T, and it is C. Okay. Now the troponin I has attached to F-actin, whereas T attached to tropomyosin and C attached to calcium. Also, the function of the calcium ion we're going to discuss in detail. Okay, so this is all about the various proteins, right? Contractile proteins. Along with this, other proteins are also there. Other protein like we have actinin, right, which attach actin filament to Z line. Then we have desmin, which binds Z line with sarcolemma. Sarcolemma is the covering of the muscle. Then we have nibulin, which runs in close association with and parallel to actin filament. Okay, so these are the other protein. Usually the question over the actinin, desmin, and nibulin doesn't come, but they can ask in the MCQ. So these are the important ones also. Okay, then other protein name are also troponin I that already we have discussed, the skeletal fast twist one, then myomycin, then myosin binding protein C, which is a slow type, the myosin light chain 3, myosin light chain 6B, then alpha actinin 2, and gelsonin. Okay, and the gene synonym are here. So this is all about the proteins. Next, we have the sarcotubular system. What is sarcotubular system? It is a system of membranous structures in the form of vesicles and tubule in the sarcoplasm of the muscle fiber. First, you should understand that if this is the muscle, then this is the covering is known as sarcolemma, and the cytoplasm is known as, do you know the cytoplasm of the muscle is known as? Anyone can answer? Fauziya, Jayendra? The cytoplasm of muscle is known as Kavya. Any idea? It is known as sarcoplasm. Okay. So the sarcotubular system is a system of membranous structures in the form of vesicles and tubules. In the sarcoplasm of the muscle fiber, okay? And it surrounds the myofibrils embedded in the sarcoplasm, okay? Now, the structures which is constituting the sarcotubular system, we have T-tubules and we have L or longitudinal tubules, okay? So, one by one, we're going to talk about both these structures. So, what is T-tubule? T-tubules are the they are the narrow tubules formed by the invagination of sarcolemma. Okay, so if this is the this is the muscle and this is the sarcolemma. So basically, T tubule are the extension. These are the extension of the membrane which get invaginate in the muscle. Okay. So T-tubule or transverse tubule are narrow tubule formed by the invagination of the sarcolemma, and these tubule penetrate all the way from one side of the muscle fiber to the another side. Okay. Now, these tubule penetrate the muscle cell through, and because of their origin from the sarcolemma, the T-tubule open to the exterior of the muscle. Therefore, the ECF, extracellular fluid, can run through their lumen. Okay. Now, talking about the L or longitudinal one. So, the L tubule or longitudinal tubule are closed tubule structure. They are closed tubule like this. Okay. And they learn in long axis of the muscle fiber like this. Okay. And forming the sarcoplasmic reticulum. Okay. So, these L tubules are basically responsible for the formation of sarcoplasmic reticulum in which the calcium remains stored. Okay. So, L tubule corresponding to endoplasmic reticulum which present in other cells, right? And at regular interval through the length of the myofibrils, the L tubule dilate to form a pair of lateral sac that are known as cister. Okay. Now we're going to discuss about the functions of T tubule and also the function of the L tubule. So what is that function of T tubule? They are basically responsible for the rapid transmission of impulse. Right? In the form of action potential from sarcolemma to myofibrils, right? And whenever the muscles are stimulated, the action potential develops in the sarcolemma and spread throughout it, and T tubule are the continuation of the sarcolemma. The action potential pass through them and reaches the inferior of the muscle fiber rapidly. Then next we have the function of the L tubule. Now, the L tubule stored large quantity of calcium ion. Okay, and whenever the action potential reaches the cistern of L tubule, the calcium ions are released into the sarcoplasm, and the calcium ion triggers the process involving the contraction of the muscles, and this process is also responsible for the or this is the cause of the excitation contraction coupling that also we're going to learn in today's class. Okay, so these are the function of the L tubule and D tubule. Now, finally, we have the role of calcium in muscle contraction. So, as we have discussed that the calcium are basically stored in the L tubule, right? And L tubule here forming the sarcoplasmic reticulum. Okay. So, what happens? Suppose this is the nerve, and here we have the muscle. Whenever the action potential reaches the nerve, so it will cause the release of acetylcholine, and the calcium which is stored inside the endoplasmic reticulum will get out, and there will be flow of calcium into the sarcoplasm. Okay, so this is how the mechanism will run. Now, what is the function of this calcium? So this calcium is going to bind over the tropomyosin, right? And what we have learned that about the tropomyosin, that this tropomyosin is basically present on the binding site of actin filament. So whenever this calcium get bind to the tropomyosin, so it will lead to the removal of this tropomyosin. And as the tropomyosin will get removed, so the myosin head can attach to that side so that the contraction can occur. Okay, so this is the role of calcium that we have discussed. So normally the head of myosin, if we talk about the head, suppose this is, we have the actin. Here we have the actin, right? And the tropomyosin is present here. Okay, so normally the head of the myosin molecule has a strong tendency to bind to the active site, right, of actin. Now, in relaxed condition, the active site of F-actin is covered by tropomyosin, as we can see, right, the myosin. So, if the active site is covered by tropomyosin, so the myosin head, this head cannot get bind with the actin molecule. Okay, now what happened, as the action potential will be there, so there will be release of calcium from the sarcoplasmic reticulum, right? So large calcium ion which are released from L tubule. L tubule is responsible for the formation of sarcoplasmic reticulum, right? Now during the excitation of the muscle, it binds with tropomyosin, and there will be detachment of this. Finally, this myosin will able to bind so that the muscle contraction can. Okay, so this is the rule. So we're going to learn it with the help of a flowchart also. So what happened whenever there is excitation of muscle fiber by or through the neuromuscular junction. So NMJ also we're going to discuss. So what will happen? There will be generation of action potential in muscle fiber. So what will happen? There will be spread of action potential through T tubule. What were T tubule? T tubule were the extension of the sarcolemma through which the transmission can be occurred. Now finally there will be entrance of action potential in cisternae. Now it will lead to the release of calcium ions from cisternae, and there will be movement of calcium towards actin filament. Okay. Finally, it leads to the contraction of muscle. This is all about the action of calcium in muscle contraction. Okay, next we have the another important, that is sliding filament theory. Okay, now the sliding filament theory explain how the actin filaments slide over the myosin. As already we have seen how the actin filaments slide over the myosin and they form the, together they form the actin myosin complex during the muscle contraction. Okay, and also this is known as ratchet theory, okay, or walk alone theory. Now, the formation of actin myosin complex. What is actin myosin complex? This is a complex of actin and myosin. When they both get attached with each other, so that time they form the actin myosin complex, right? Now, one by one, we're going to learn what happens whenever there will be release of calcium and calcium get binds with the tropomyosin and tropomyosin will get removed from the active site. So what will happen? The head of the myosin will get attached with the actin filament, right? Now, at that time, there will be removal of ADP and inorganic phosphate. So always remember, at the resting stage, the ADP and inorganic phosphate always remain attached with the myosin, whereas whenever it get attached, the head of the myosin filament get attached with the actin, so they both get removed, right? So once the cross switch form, the myosin perform a power stroke. What is power stroke? Power stroke basically when each cross bridge formed from the myosin filament has got the three components already we have discussed. So whenever it get binds with actin filament, so that time it is producing the power stroke. So in short form, we can call the tilting of head of myosin is known as power stroke. Okay. So once the cross bridge form and the myosin perform power stroke pulling the actin filament, so that time the ADP and inorganic phosphate are released. Okay, and as the contraction will occur, so new ATP attached to myosin and head and cross bridge detaches. Okay, so new ATP will here will be there and this crossbred detaches. Now the myosin uses ATP to return to its original position again. So with the help of this ATP will be a hydrolysis and again it converted into ADP and inorganic phosphate and again it will get relaxed. So with the help of the attachment of the myosin head, it leads to the sliding filament theory. So as the myosin head will get attached like this, so both the actin filament will contract like this. If suppose this is the actin filament to actin filament and this is the myosin. Okay. So once the myosin head get detached with the actin filament, the both actin will slide over each other and causes the contraction like this. Okay. So next we going to learn again with the help of the flow chart. So whenever there is release of large amount of calcium from which tubule? Anyone can answer? From which tubule there will be release of calcium? L or T? Kavya, Nikita, Fauzia, anyone can answer? From which tubule? So the release of calcium will take place from the L tubule, right? Now there will be binding of calcium with troponin. And as the calcium will bind, there will be change in position of troponin. Right? Now what will happen? There will be exposure of active site of affectin and attachment of myosin head to actin, right? and it will lead to power stroke. Power stroke means tilting of myosin head and dragging of actin filament, right? So it will lead to power stroke, and there will be again detachment of myosin head and return to original position. Now what will happen? There will be sliding of actin filament and contraction of muscle will occur. Okay, so this is all about the sliding filament theory. Now, next we have the another important topic, that is NNJ. NNJ means neuromuscular junction, means the junction which is present between nerve fiber and the muscle fiber. Okay, so NMJ is also a very important topic, and also we going to not we going to discover discuss it in detail, but we will have a very quick recap about it. Okay, so as we know, this is presynaptic membrane and this is postsynaptic membrane. Okay, and the space between both presynaptic and postsynaptic is known as synaptic cleft. Okay, so what happened as the action potential which reaches the terminal part of the nerve, so what will happen? There will be opening of the calcium, opening of the voltage gated calcium channels, right? As there will be opening of the calcium gated voltage channel, so this calcium will get released and there will be entrance into the nerve. Now what will happen? This will get attached, this calcium will get attached with the vesicles which is containing acetylcholine. Okay, as there will be, this vesicles will get attached with calcium, so this will lead to here, they will attach to here and they will be released to acetylcholine. So as the acetylcholine will release into the synaptic cleft, so what will happen? This acetylcholine will bind with the receptor which are forming the acetylcholine complex, and they will be opening of ligand gated sodium channel. As the ligand gated sodium channels will be open, there will be entry of sodium ions from the ECF, and there will be development of EPP. What is EPP? EPP is end plate potential, and various end plate potential will finally able to produce the action potential, and this will leads to muscle contraction. So what happened? Whenever action potential is there in the motor nerve fiber, so what will happen? There will be opening of voltage gated calcium channels, right? So as they open, there will be entry of calcium ions from ECF. Now, opening of vesicles and release of acetylcholine to synaptic cleft. Okay, now what will happen? This acetylcholine binds with receptors, and which receptors are present? These are nicotinic receptors. Okay, as they will bind, they will be opening of ligand gated sodium channels. As there will be opening of sodium, the ligand gated sodium channel, now what will happen? There will be influx of sodium ions from ECF, and it will lead to the development of EPP, end plate potential, and summation of various EPP will lead to generation of action potential, and it will cause the muscular contraction. This is all about the NMJ. Okay, next we have the technique. So as already we know, what is tetany? Tetany is the abnormal condition, right? This is the abnormal condition characterized by violent and painful muscular spasm. Right? So, but basically, particularly, it occurs in hand and feet also, and it is because the hyperexcitability of nerve and skeletal muscles due to calcium deficiency. So, the main characteristics will be that it occurs due to calcium deficiency. Okay, so this is the involuntary contraction of muscles which create cramp, spasm, and sensory disturbance. The classical sign of hypocalcemia that there will be decrease in the resting membrane potential of neurons and increase in the neuromuscular excitability. Also, there will be combination of metabolic abnormalities and hypocalcemia. There will be hypothyroidism, vitamin D deficiency, increase calcium leaving the blood, and there will be electrolyte imbalance disorder affecting the calcium acid base balance. Then the symptoms, usually the numbness, painful muscle cramps, paresthesia, then laryngospasm is there, then vomiting, seizures, and decreased cardiac function are the main features of the technique. So another characteristics or sign and symptoms also we can write. Carpopedal spasm. What is carpopedal spasm? Basically, this is the spasm that occur in hand and feet that occur in the condition like hypocalcemia tetany. Okay. Then various cardiovascular changes are there. In which there will be dilation of the heart, prolongation, duration of ST segment, right? Arrhythmia, hypertension, heart failure, okay? And other features are also like dry skin, hair loss, decreased permeability of the cell membrane. So these are the other signs and symptoms of the technique. Okay. Then finally, coming to the disorder that is caused by the NMJ, and that is myasthenia gravis. Okay, so we're going to learn what is myasthenia gravis. So again, the myasthenia gravis is a autoimmune disease. So as already we know, what an autoimmune disease? Autoimmune disease are those disease in which our own body creates the antibodies, right? So myasthenia gravis is basically due to the development of auto antibodies against the receptors of the acetylcholine. So as we have discussed, these are the receptors for acetylcholine. Over the acetylcholine will bind, but in myasthenia gravis, there will be formation of antibodies against the acetylcholine receptors. Okay, and these antibodies prevent a binding of acetylcholine with the receptors or destroy the receptors. So the acetylcholine release is normal, but it cannot execute the further action. Okay, now what are the signs and symptoms? So we have the signs and symptoms like slow and weak muscular contraction, then weakness of arms and legs, difficulty in swallowing, difficulty in speech. So these are the main signs and symptoms or the problem which occurs in this disease. Okay, so this is all about the NMJ disorder. Then finally, already we have discussed the role of calcium in muscle contraction. So we know the muscle are responsible for removing the tropomyosin over the active side of the actin filament, and so that the process of whole muscle contraction can be occurred. So if there is anyone who will be able to draw the whole sequence that already I have discussed with you, if there is anyone? Who can tell me to describe the role? Anyone? Fauziya, Kavya, Nikita, Jayendra? Okay. So whenever whenever there is release of calcium ions from cisternae, this will lead to movement of calcium towards actin filament, and finally it will lead to the contraction of muscle fibers, right? So this is all about the role of calcium. Then the sliding filament theory, already we have discussed what is sliding filament theory, in which there will be release of calcium it binds with tropomyosin, removal of tropomyosin from the active side, myosin head binds with actin, producing the power stroke, and there will be dragging of actin filament, and finally this will lead to sliding of actin filament of both sides. So in just short, I have written, but if the question over the sliding filament theory will come, so you have to draw the whole chart like this, and also you should draw this diagram. This is the most important one, and also you need to explain that what is the muscle protein, what are the muscle of proteins are present, what is sarcomere, what is sarcoplasm, what is sarcoplasmic reticulum, all these structures that we have discussed in today's class, all you need to explain in detail whenever the question over the sliding filament theory will come. Okay, so this is all about the today's class of muscle physiology. I hope you find this class more helpful. If you have any question, so you can contact me through the various through YouTube, through Instagram, or through various modes, okay, that Auricle has provided. So thank you so much.